Hello, everyone, and welcome to the DNA of Things, where we explore how our potential is realized at the intersection of DNA, environment, and behavior. Here, we unravel the stories behind cutting-edge genetic research, transformative health insights, and the people pioneering a new frontier in health, human performance. Dr. Chris McMaster is my guest today. He's a geneticist, entrepreneur, former Dean of Medicine at Dalhousie University, and as the scientific director of the Canadian Institute of Health Research and the Institute of Genetics specifically, he's shaping the future of gene therapy, rare disease treatment, and collaborative science. Today, we dive deep into topics such as how gene therapy is shifting medicine from treating symptoms to curing disease at the DNA level, the power of collaboration in science and why competition often slows innovation, and the ethical dilemmas of genetic and genomic medicine. Who owns your DNA? And should newborns be sequenced at birth? This episode isn't just about the science, it's about the future of medicine and what it means for you. So let's dive in. Dr. Chris McMaster, extensive experience coming from his role as a scientific director at the Canadian Institute of Health Research, Institute of Genetics, his groundbreaking contribution to orphan disease treatments, pharmacogenetics, and leadership in drug discovery. He has been entrepreneurial for as long as I have known him. His most recent venture, Allogene, and his collaborative work in genetic solutions for muscular dystrophies and infectious disease are building a path to a more hopeful future where individuals can understand their bodies and empower health and health span. Dr. Chris McMaster, thank you so much for joining me today on the DNA of Things.
Thank you, Jeremy. Of course, call me Chris.
Chris, right. Well, I mean, this is, I would say, what would you call it? Me coming back to the beginning or where I came from and seeing it for the first time, right? So I mentioned in our last conversation, I was reflecting on the fact that I took your advanced biochemistry labs on. I think you were a site-directed mutagenesis, if I am not mistaken. So if you can believe it, it's been 20 years since.
Yep.
So I'm delighted to be reconnected and, learn, how much you've changed, how much the genetic space has changed. But, as I mentioned at the outset, in order to do that, I've taken this hero's journey format where you're the hero and it's your journey. And I want to learn a little bit more about you. Listeners are going to want to learn more about you in terms of your journey into genetics and innovation. So what was it that captivated you by the industry, and what were some of the kind of key moments that have pulled you along to where you find yourself today?
Thanks for that generous intro, Jeremy. I would say in terms of key moments, I started out as somebody who was using genetics just to try and figure out how our cells work, and basically functional genetics. I was disease agnostic. I really wasn't working on a disease. However, I was planted in a department of pediatrics for the first 15 years of my career. And it became very clear to me as genetics and genomics progressed that there was a window where we could now start to apply some genetics and genomics technologies, just because the price of sequencing a genome dropped so quickly and the speed with which you do it sped up so quickly. We basically went from the first genome being over $1 billion and taking a decade to now in my lab, I can sequence your genome or my genome for a few $100s in an afternoon. And so when that capacity started to become available, there's a lot of kids that are born with a genetic disease. So out of the 21,000 diseases in us, the genes in us, one different gene has gone sideways in these kids. They've done nothing wrong. They're just born with this disease, and it's a big number. It's 1 in 20 kids born. That's a genetic disease that results in getting sick and often die before they reach adulthood. And I just said, well, there's two issues that are big problems for the parents and the children themselves. The first was the speed to diagnose these kids was still clinical. What do you look like? What's your disability? What's your blood work like, et cetera. It would take 5 to 7 years. Pile of specialists, 20 different diagnostic tests, a couple misdiagnoses, and they'd finally get around to getting a diagnosis. But in the age of genetics and genomics, we've now been able to speed that 5 years and tens of thousands of dollars. You can imagine being a parent and your kid is failing in front of you physically and or mentally. And it's this 5 to 7 year journey to get to a diagnosis. Let's try and make that 5 weeks or less using genetics and genomics technology. So a lot of what we've been working on is to try and discover genes that when they do go sideways, cause an inherited disease, and then incorporate that into the clinic so that you can decrease this diagnostic honesty that the families and children suffer through from years to weeks. And so that was the biggest push in the beginning, was just it could be done. So let's just start doing it and do it at a local and now national need.
Wow. Just a quick distinction there, genetics and genomics. You're very careful to use each of those as different terms. Can you describe the difference between the two?
So our genome is essentially, we've all got the 3 billion letter code, A, G, C, T, which are the 4 bases that make up DNA. And we all have 3 billion pairs of those, that if you add it all up, that's what we are. And so we can now sequence all 3 billion in a person. Now, that's your genome. The genetics is then, okay, how does your genome affect your life? Is there anything within your genome we can take a look at that affects your capacity to stay healthy, your predisposition to disease, your predisposition to side effects if you take a certain medication, your capacity to metabolize different nutrients or not? There are a lot of things we can learn from your genome that when you look at it sort of from a 30,000 foot level, your genetics is sort of the phenotypes that your body then has to deal with based on your genome, which is just the 3 billion letter code that comprises all your genes linked together.
Yeah. So I want to come back to some of the newborn screening initiatives that are currently underway, maybe toward the end, because I know we spoke about those. And, I think the good news is there has been, massive advancements since you started, and there are a number of pilots underway. And, newborn screening might be, not just for the 1 in 20, but it might be for all of us proactively in the future. But maybe before we kind of jump to that, hopeful future, I'd love to learn, a little bit more about, how you've been able to balance the academic and the industry side of things, because, besides being in the field for, well, let's just say many decades, you've, held positions academically. I believe now you're currently, you're a professor in the pharmacology department, but I believe you're the dean of medicine as well at Dalhousie University. That's correct?
Well, I was an assistant dean at Dalhousie University for 7 years, and I was the head of the Department of Pharmacology for 7 years, and I've been the scientific director at SACHAR, which is the largest funder of research at universities and hospitals, for 7 years as well. So it's 21 years of sort of balancing some sort of leadership job with also running a research lab at the same time.
Right. And you actually have your own ventures as well. So
there's been two spin-off companies, like you mentioned earlier, Jeremy. One was in the infectious disease realm, which we won't bring up today. And the other is a recent venture, Allogene Inc., which is in the gene therapy realm. Part of this is serendipity. It turns out the genes that I worked on way back when I was a PhD student, postdoc, in early days as a professor, they were all thought to be essential genes that if you inactivated them, and they did it in cells and in the very first mouse, nothing survived. So they said, oh, they're essential. It turns out that for every step in this pathway, instead of there being one gene, there's 2 genes for every step, which we learned from genomics. And as people were sequencing lots of genomes, and a variant in any one of these genes actually causes an inherited disease in kids that's a neuromuscular disease. And one of them's a muscular dystrophy. So if you just happen to inherit variants in this gene that cause it not to work, the child is born with a muscular dystrophy that progressively gets worse, and they will usually die in their 20s or 30s. And so there's a muscular dystrophy expert who also spun out a company called Agata Biosciences here in Halifax, that is a contract research organization that does a ton of work on neuromuscular diseases, and all the preclinical work required to get a therapy, including gene therapy, is approved. So we figured, well, I worked on this pathway for 30 years. The CEO of that company has worked on muscular dystrophies for 30 years. His company is crazy successful in helping others move therapies into first human trials. Let's work together to move a therapy on this muscular dystrophy that I've been expert on, I would say, for 30 years now, towards the clinic. If we can't actually get it done, we should just both quit and retire. So that's our plan.
Well, I mean, maybe you're being a little too hard on yourself because you have the expertise in terms of the development of the therapy. You have the expertise in terms of the biochemical pathways, but it's no small deal to coordinate a clinical trial, is it? In terms of, qualification and adherence and efficacy and all that. I mean, how do you coordinate all that?
I think one of the reasons I've been able to research lab and have these side projects, let's call them these other ventures taking place at the same time. It's a leadership job. The spin-off company is I've been very fortunate to work with and hire excellent people. And that's really been a huge key to my success is I am not a micromanager, horrible micromanager. I'm very macro when I manage. And so I give the advantage, I guess, and disadvantage if you end up working with me is that you get a lot of freedom, but you're expected to be very independent and be able to pull your own weight. And I've been fortunate that the folks I've worked with have been able to drive a lot of projects forward. Of course, we talk every week, usually several times a week, but they've been able to steer the projects and actually own them as much as I did when it comes to, getting from the beginning of the project out to, the end of the project and over many, many projects over many years. And so having wonderful people to work with, first thing I look for when I'm hiring somebody is, can they play well with others? Can they get along? And first and foremost, and then, do they have the qualifications for the job? And are they comfortable working independently or not? Because you need to be comfortable and want to work independently if we're going to move things forward in the way I tend to move things forward. And so I've had wonderful staff, both on the, let's say, bureaucratic side of my life, and I've had wonderful staff on the scientific side of my life. I guess one of the nice things about the job I've had is that I get to hire the staff, and so I can choose. And I've been lucky that I've hired some excellent people, and that's one of the biggest helps that I've had all over. It's just wonderful people to work with. But I also seek out wonderful people to work with. I don't know what kind of language policy you would have for this, but basically no assholes allowed when I come to choosing people to work with or not. I just don't have the patience. Won't do it.
It's funny, what you just described as your leadership philosophy actually echoes Ford Doolittle. You probably remember him. He was a neighbor of yours in the Tupper building there. Yeah. And I remember starting my graduate student work with him, and he said, look, I think you're smart and you should join my lab. But here's the thing, I'm going on sabbatical and you're just going to have to figure it out. But it was like, here, by the way, here's all these great resources. I mean, when I joined, the lab was booming. I think he had between 8 to 12 postdocs. So it wasn't like, I was going to have to figure it out all by myself, but, certainly I was expected to drive my own project, but there, as I say, no shortage of resources and, great minds to support the process. And it turned out to be like, at first it was really hard, I compare it to going into a med school degree where it's like, here's what you have to do, at least for the first year or two. Like, here's the coursework and the study plan, and here's when you'll be examined, and here's when the competition is, and here's when you need to be ready. Versus, with grad school, it's kind of like, yeah, it's a very loose structure. Like, sure, you have committees and you have, specific competencies that you're supposed to demonstrate in terms of, your research project. But what is your research project, and is it a good one, and is it a question that is worth asking? And so, I mean, that took, probably a year, 18 months to even figure out what the question was. And, of course, it wasn't doing nothing the entire time, but, certainly I was afforded the time and space to make mistakes, which was great. But in the end, there was a great, kind of, pursuit of, well, you might call it one of these creative destruction labs where it was really, deconstructing Darwin's tree of life, right? And then rebuilding it back up into a new paradigm of, understanding evolutionary process. So even that was challenging because it's like, oh, wow, everything I learned in undergrad was, well, not wrong, but it's being challenged. And I think that's something that I didn't really appreciate until I started actually doing research, is that, the answers aren't there, and it's kind of your job to find them, and you don't. And then, oh, well, that's why it's called, research, on that. Like, as you pursue this career, I would say, it's probably no surprise that you have a fair degree of, it's no surprise that you then go from your research world to be an entrepreneur, because I think there's a lot of overlap between being a researcher and an entrepreneur insofar as the uncertainty. So just through that journey, what are kind of some of the highs and the lows for you? I mean, it could be, entrepreneurial or academic, but is there a moment that comes to mind that was, just exhilarating? And then was there another one that was just like, wow, that's my low of lows throughout this journey. Can you remember anything like that?
Oh, sure can.
Yeah.
There's a few. We'll start with the low of lows, I guess. One of the things about research, especially when you're brand new, coming out of your bachelor's degree and you're starting your PhD, is that you go from a very structured environment where here's some assignments, here's some tests, here's some due dates, to a PhD where you really set your own agenda and your own ambition. Because it is exactly that. You show up when you show up, you work as hard as you want to work as hard as you feel like, and you work as smart as you feel like. And at the end of the day or the end of the week or the end of the month, what have you got accomplished? And I, in the first couple of years of being a graduate student, really struggled with setting my own agenda. And after a couple of years of grad school, my supervisor sat me down and basically said, I don't think graduate school is for you. Really are a little too all over the place in terms of your work ethic, in terms of your thought processes. And unless you can get these things organized, you're never going to succeed in this job. Maybe you should just write up your master's and think about doing something else for a living. Thing is, I really liked research. And so I thought to myself, well, geez, you got to get your act together here, McMaster. And so I actually had to learn. And it was a learning process for me to actually find my own, not ambition, but find my own process by which I could work in this very sort of loosey-goosey environment. Because you're the only one who sets when you show up, how hard you work, how accurate you work in terms of you doing bench work. And it was really a matter of me putting my nose to the grindstone and learning how to do that well. And so I just said, look, can we meet more often? Because he was, again, a guy who was a department head and president of the Heart and Stroke Foundation, and he had a lot of stuff going on as well. I said, at least for the first few months, could we meet more often so I can try and get on track a little better? And he said, of course. And so we did. And then within the next 2.5 years, so in 4.5 years total, with the first 2 years being a bust, that was defending my PhD. And when I applied for the CIHR fellowship, I got ranked first in the country. So I did find a way to get that done, but it was hard. It was a big learning curve. The second place was, and this happens to a lot of people, you get your job as a shiny new professor. And once you're a shiny new professor, you've got all the modern techniques and all the modern everything in your CV looks pretty good in terms of what you've published, et cetera, et cetera. And so your ideas are usually pretty, cutting edge. So getting your first grant really isn't all that hard, but it's not all that. So I got a couple of CIHR grants, an insert grant, the heart and stroke grant, flying high. And then 6-7 years into my career, the 2 CIHR grants did not get renewed. The heart and stroke grant did not get renewed. And I was left with an insert grant, which is the smallest of the bunch. So I applied for renewals. And the success rate for most of these grants is about 20%. And I kept sitting at about 25, 26% and couldn't get past that number. And I went, oh, man. So I went and talked to some folks around the medical school. Ford was one of them. Jerry Johnson was another. And said, all right, what's the problem? How can I make this grant better? And they said, I don't think you can make the grant better. I think you're just topped out at 25% because that's the quality of the idea you're pitching. Damn it. And they said, so you're going to have to change it up, pitch better ideas. And I didn't know what to do. And so that was where I went from working as sort of a basic scientist using genetics to try and figure out basically the biology of a cell and a lot of that was, there's enzymes that convert metabolite A to B to C. I was figuring out the genes in the enzymes that did this, characterizing enzymes. A lot of them are inhibited by their products, the end product of tablet, which is a small molecule. And I said, well, drugs are small molecules. Maybe I could find somebody that's good at making drugs and designing drugs. And instead of working on what's going on within the cell, we could actually find drugs that inhibit enzymes that are important in disease and pathways I know something about. And that could actually turned the corner for me in terms of better ideas for research. That resulted in me working with a guy called Donald Weaver. He's a computer-aided drug design expert to Canada Research Chair, yada, 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 very impressive guy. And I pitched him an idea, which was, I worked on metabolism, not only in people, but all the way down to bacteria. I said, if we shut off this enzyme, bacteria, they can't make membranes and they'll die. It's essential in how they can serve. And it's a very small protein with a defined active site where we could put drug into. What do you think about working together? He said, sure. And that turned the corner for me big time. And was my first foray into getting a startup company up and running from university-based research. But he'd done it before, which was nice. So he was able to just basically show me how it was done. So again, seeking out mentors, people that are brighter, some usually older from me, smarter than I was at the time in terms of being able to accomplish these things. And that made a big difference. So those were the two low areas that they then had to sort of. One was changing my attitude early on and my work ethic as a young and early 20-year-old. And then, as a not early-ish, but 7-8 years into my academic career as a professor, I had to change my research approach because I topped out in terms of how important what I was pitching was doing, never get funded again. So I had to change gears, but not a lot. It was changing from small molecules of endogenous that inhibit enzymes to finding somebody to help design drugs do the exact same thing. And that was the antibiotic company, the Novamid, that spun out. And I've sort of along those lines now. And now we're working on inherited diseases in kids. Again, defects in lipid metabolism that cause these kids to get some sort of inherited disease. And the best in class way to actually fix these kids now is gene therapies. And so I'm working with folks that are experts in gene therapies and models of gene therapies and have actually got products approved by the FDA in the past. So again, you can't do it all on your own. It's too big a space. And so you got to find nice people to work with who are collaborative, who play well with others, and have the mission of there's people that are sick and you want to do something about it. So it can't be about you. Because if you make it about you, one, it's no fun. It's far more stressful. But if you make it about the patients and the parents, in this case, the kids, it gives you a much bigger sense of purpose and a bigger reason for you to want it to succeed versus making it about yourself. So I think for a lot of researchers, I think they forget the end game sometimes, which is these are kids that are sick or adults that are sick. And the end game isn't to write more papers and get more grants and be able to travel to more conferences and present your work. The end game is there's people that are ill and you're trying to do something about it. And so it's not about you, it's about them, and work towards that is your goal.
Okay, a couple of things. I mean, I first thought on, what you just described there, like the end game or, the North Star, if you will, versus the KPIs, right? Like it is, and I like to use like the, having a bit of a background in martial arts, boxing in particular. It's like, okay, well, your goal is to win this tournament, and part of the KPIs are going to have to do a certain level to your technique and your strength and your capability, and you're also going to have to make weight, right?
Yeah,
it's pretty easy to make weight. You could probably do it in a week. It's not really a good indicator of if you're going to win, right? And it is, I think it's tempting isn't the right word, because I don't think people are tempted by the KPIs. I think they're genuinely going after the North Star. But, the fact is, like, you got to keep the lights on. Well, how do you keep the lights on? Well, you get grants. Well, how do you get grants? You publish, right? So, it's kind of part and parcel to, the system itself, right? It is okay. Well, how do the granting bodies define success? Well, I think that has a lot to do with the administration. What's their focus on? Is their focus on basic research? Is it based on applied research? So there is like this whole kind of, bigger frame that you need to navigate. I think you're right. You kind of collect good people who are focused on that end game, because no one would refute that the real goal is to decrease pain and suffering in the world, right? So I guess, like, how do you surround yourself, with people? And I think you told us at the outset, it's like, well, that's kind of been your priority. You indexed on the other side of it, i.e. no assholes. Well, the extreme opposite of that is, people with hearts of gold. So I wanted to come back to one of the things that you said at the beginning, which was, well, let's call it progress as it relates to technology. So you mentioned specifically at the outset, the cost of sequencing. I seem to recall you were part of a group that I was a part of in my undergrad working with Melanie Dobson, and that was like the yeast group or the yeasties or whatever it was they called them. So we were using yeast genetics and specific functional assays that was probably related to your small molecules project, if I was guessing. But what other, and you did mention when you started, there's all this cool, shiny tech and, you're, the golden child, but it's probably changed a lot since you've started. And so what's changed and has that changed, how you go about doing science today?
It's changed so much. The reason I was starting out in yeast genetics was it was the only organism where you could easily knock out a gene or knock in a gene because they're diploid and haploid, and the genetics in terms of yeast liking to recombine into their own genome was highly efficient and effective. And so if you wanted to look at a eukaryotic cell, be able to do genetics, it was by far the fastest, most cost-effective eukaryotic cell you could do that in. You couldn't do it in mammalian cells, so there's no technology that allowed you to do that. And then about 10 years after me becoming a faculty member, so in 2010-ish, Allogene came along, which were a way to knock genes out in mammalian cells, but they weren't super effective. But then CRISPR came along, and it works great to be able to knock out and knock in things. And so now I do very little yeast genetics in my lab, and we've moved on to CRISPR to manipulate human cells. We've also moved on to using stem cells from patients, which is, you can get a skin cell from a patient. You can de-differentiate it into you can get a fibroblast skin cell from a patient, de-differentiate it into a stem cell, and then re-differentiate it into whatever tissue is going wrong in that person. And in inherited diseases, if it's a muscular dystrophy, you re-differentiate it into a skeletal muscle, but if it's a cardiac problem, you re-differentiate it into a cardiac myocyte. Then you've actually got a cell type that is got that person's genes, but it's in a cell type that's relevant to the disease versus a skin cell. So stem cells have really changed things as well. And then if you want to work on the etiology of the disease, or if you want to test a therapy or screen for therapies, you can do it in the exact cell that's from that patient that makes sense now. Starting from a skin cell, going backwards to a stem cell, and then back forwards to a muscle cell or a brain cell or neuron or whatever. So that's changed as well. But also, you need mouse models of disease. And inherited diseases, it's easy because they're single gene disorders. So you can knock out or knock in the gene, see if your mouse model is a good replicate of the human gene disorder. And if it is, you're off to the races. Fibroblasts, skin fibroblasts from patients that you can then de-differentiate to stem cells, re-differentiate it to actually skeletal muscle cells for a muscular dystrophy. And you can work on those. You've got a knockout mouse that's got the same gene knocked out as these kids have. It almost perfectly recapitulates the disease. So you get another way to determine disease. You have etiology and test therapies, in our case, gene therapies. And all these things used to take forever to do and cost tons of money, or you couldn't do them at all. And in the last 10 years, all of these things in terms of doability and cost have just plummeted. And so the technologies are now there to take a really good run at a lot of inherited diseases, most of which affect kids, by just using skin fibroblasts into stem cells, into whatever cell type is gone sideways, and using mice that are either knocked in or knocked out with the genetic change that is present in these kids for disease models. And with gene therapies now existing, where you can actually take, for recessive genes where things have actually, the gene doesn't work, putting in the right gene, it's also not rocket science anymore. It's way easier than a drug to actually get over the finish line, show that it works. And then if it does work in a mouse, the odds of it working in a human are actually quite high. So and that's just super recent, the last 5 years. The technology is there. All of those things have just accelerated so quickly that, for example, the cost to get a drug over the finish line, if nothing goes wrong, it's about $100 million. Cost to get a gene therapy over the finish line, if nothing goes wrong, is about $12 million.
Wow.
So it's a doable thing for a lot of these kids. And because these diseases are quite rare, it's really not a space big pharma is looking at to make big bucks. And so most of the folks that are taking the run at these are academics who are spinning up little biotech companies like us. But you don't have to raise $100 million. You just got to raise 10 to 12 million. You can probably have it approved and ready to go. And so it's a doable thing for a small biotech to be able to accomplish.
Okay. So you're focused on muscular dystrophy to start. So okay, I don't want to future pace too hard. Obviously, we're focusing on getting a win there, but if you prove the model, is it kind of like wash, rinse, and repeat for more of these kind of rare disease, underserved populations, which there's 1 in 20, right? So it's just, they're so disparate and fragmented that it's hard for big pharma to make a business case to go after it. So is that kind of where you're thinking?
We're thinking of trying to platform it a little bit because there's over 30 different muscular dystrophies, 30 genes that make up muscular dystrophy. And some of them are more amenable based on scientific tractability to a gene therapy than others.
Right.
You need a gene that's relatively small. You want your protein to probably either be soluble or excreted. You don't want it to be an integral membrane-bound protein or something that needs to get in the mitochondria through a signal sequence. All sorts of things can make it harder to do. You don't want a protein whose overexpression is toxic to a cell because it usually overexpresses things. So there's some scientific pieces you got to check out first, but many of them are tractable. So we're going to take a run at probably a half a dozen of these. And because we're trying to correct the muscular dystrophy and skeletal muscle, once we land on our gene therapy backbone for it, which we've pretty much done, you can just cut and paste your gene of interest, and then your development cost after the first one is over the finish line is quite different than the second, third, fourth, fifth, and sixth, because you through that it works already. Just switch another gene.
Yeah. Right. Okay. And so up the assembly line from there is it's the DNA analysis. So are you going back to like the whole genome sequencing? Because maybe some of these you don't know a priori, right? Because they're undiagnosed. A lot of these are rare undiagnosed, I'm assuming, right? Or are these rare diagnosed? Are you going after like kind of established markers, or are you discovering at that point as well?
Yeah. So for the ones we're taking a run at, they're either people who part of the discovery team that discovered the gene. In our case, the one we're going after, I was part of that. I've discovered a few more that will be follow-ons to our first project, but then we're talking to other folks who either discovered or were in the lab that discovered the gene that causes the muscular dystrophy. So they've already got deeper expertise in this. So we're going after genes that are already known to cause a muscular dystrophy. They're already on the panels that if a kid shows up thinking they have muscular dystrophy, well, this is one of the 30 genes. It's already on the panel. So we can find patients quite easily because they're on all the gene panels that anybody who has muscular dystrophy is going to have looked at.
Mm-hmm. Mm-hmm.
There are still a bunch of genes that need to be discovered. Out of the 7,500 rare diseases, we're already up to about 5,500 genes. So that's really good hit rate. So we're already past 2/3. We're hitting, there's another 2,000 to go. We're still working on gene discovery efforts for the other 2,000 as part of our research. But now that we've got a whole plethora, 5,500, it's a lot. So there's a lot of hay to be made. That's probably not the right word. But there's a lot of work that can be done now. And now it's just parsing these into, for me, scientific trackability for these 5,500.
And so how were those 5,500 discovered initially?
It's either through one of two ways. You either have a pedigree where you've got a lot of family members and some have the disease and some don't. And then you sequence their genomes, and if you can sequence enough genomes, you can say, okay, the disease tracks with these variants. It does not track with these other genetic variants or genomic variants. And that leaves us with these 3 or 4 genes that keep tracking with the variant. The bigger the family pedigree, the better.
Right.
And then if you can actually find some people that have the same disease that are not part of that family, they'll have less genetics, less genomics shared with the family. So that can really start to eliminate some of the other variants that are shared through families. And so if you can get another 2 or 3, even single individuals that have the same disease, sequence their genome, that makes it a lot easier. Genomics algorithms to say, okay, it's either this gene or this gene. It has to be.
Got it.
Bit of a mass action effect, as you can see from that.
Yep.
or even 10 years ago, a genome was $50,000. It's like $8,000. So it's really been in the last 6, 7 years, 5 years, where we've just been able to algorithmically discover all these genes that cause these inherited diseases.
Yeah, it kind of sounds like like the BRCA1, BRCA2 story with the Mormon population, like on steroids, right? So
It is.
So you've got because even I think before with like the breast cancer screens, you had to know I think you had to know something about the function of the area that you were targeting a priori so that you could focus on a subset of the genome to be sequenced. Let's say like you're using like maybe kind of PCR approaches to like conserve regions of the gene of interest. So how much has because we didn't even talk about like bioinformatics and obviously as an extension, the AI component to this. I mean, I'm probably going to say something like, AI and you're going to probably be thinking, yeah, we've been using AI way before GPT or like, so has that affected, your, kind of workflow with like a lot of the changes that we're seeing in the AI industry in particular?
Yes and no. I will say in terms of gene discovery, AI still hasn't played, disease gene discovery. It hasn't played a large role. It's still sequence a bunch of genomes of this people with the same disease, just compare them and find the variants that track with the disease and exclude the variants that don't. And doing that usually works just fine. You get your hands on enough genomes. And since sequencing genomes is so cheap these days, it's just finding patients with the same disease is probably the rate determining step. The bioinformatics is still not super straightforward. It's still a hard job. And I will say the rate determining step in terms of finding disease genes is not the sequencing of genomes anymore, which it used to be. It's now the bioinformatic analysis. But part of that is we don't have enough bioinformaticians and computational biologists just because the speed and cost of genomes has decreased so quickly that we're just saturating the people that we have with so many genomes that they don't have enough time in the day to get through them. So we need to increase capacity, Canada and elsewhere, of people with this kind of expertise. I think where AI is going to cut, it's going to be helping with the diagnosis of somebody who shows up in a clinic and usually a kid, and they've got a set of symptoms that are quite doctors, family physicians. The reason they're rare is they're rare. Less than 1 in 2,000 people that show up, and usually far less than that. 1 in 10,000, 1 in 100,000 people.
Right.
All combined, it's 1 in 12. But each individual disease is incredible.
Yeah.
So where the AI is going to help is, okay, somebody shows up to a doctor's shop and they start to become, kids with rare diseases are frequent flyers of the medical system because they're all having something wrong with them. So they'll show up to their family doctor, get referred to neuro, they'll have an emergency room trip. The neuro won't be able to figure it out. They'll get referred to endocrinology or developmental peds or something like this. So they keep pinging. And AI can start to pick these kids out from medical records. Right? The frequent flyers of the medical system and why they're frequent flyers of the medical system. And they can pull those kids out and say, this kid probably has an inherited disease. So let's sequence their genome, as soon as possible. And so that's one way it's going to help. The second way is a bit of a clinical/research project, and they're doing it in England now, but I anticipate in Canada and most of the rest of the world, when a child is born, 7 or 8 years from now, they'll have their genome sequenced at birth. And right now, you get a blood, every kid gets that little heel spot test.
Yep.
Basically, what that's doing is it's looking for 20, 30 rare diseases. But the rare diseases we're looking for from that test hasn't changed in 40 years. Because we're just using the same old-fashioned genetic and biochemical tests on that blood spot that we've used in 40 years. Well, since we can sequence a genome, why don't we just look for all 5,500? Now, they're not all red light, green light. If you have this variant, you're definitely going to get this disease. And if you don't, you're definitely not. But many are red light, green light, and there's a few yellow lights in there where you might be predisposed to getting this disease. But what that does is, even if you as a person don't want to know that about your child, at least your doctor will know that I should keep an eye on this kid for these various symptoms, because I know based on genome, they could potentially be going down this road to this inherited disease. And so it gives the doctor sort of the inside scoop of either, yes, you definitely have it, no, you definitely don't, which is the majority of genes, and then a small subset that are genetic yellow light. You might get it, but now I know to keep an eye for this so that you don't end up then going through the diagnostic odyssey of, well, this kid's got some weird symptoms going on, and you do the 5 to 7 year diagnostic odyssey that most of these rare disease kids go through, 20 different tests, 3 different diagnoses. They finally get around having a genome done. Yada, yada, your genome will be seen at birth. So we'll be almost prognostically giving the doctor some information. And as we sequence more genomes and follow people's health longitudinally, if we're doing that for every kid that's born, accuracies for predicting will just go through the roof as well, because it's a data-driven, big data-driven, mass effect type of a thing. So give it 20 years from now, and it'll be incredibly accurate for all the rare diseases, the variants, and everything else. Right now, we're pretty accurate, but it's good enough, I think, to be worthwhile starting now if I was in charge of it. But I think it'll take 7 or 8 years for all the powers that be to get around to thinking it's a good idea. I guess the other problem is in infrastructure. We don't have enough sequencing machines in our clinical labs and enough bioinformaticians in our clinical labs. So we need to increase that capacity.
Right. Well, and then there's like kind of the high touch element, right? The genetic counselors. It seems like you talk about, bioinformaticians being in short supply. I mean, genetic counselors being in short supply, I think is that only exacerbate the situation, I think, right? So I guess like that's this kind of, ethical and educational challenges that we spoke to before. There's well, it's interesting the way that you kind of circumnavigated that there. You kind of smuggled that in either just by accident or by intention, but it's like, well, you don't have to know, but your doctor knows, right?
You can choose to know or not. And if you choose
yes.
It's like any other diagnostic thing they're going to do to you. You send your blood work out and it comes back, and you don't need to know what your blood work says. Most people don't, even if they look at it, don't understand what it means. But they're not going to know what your blood work says, and they'll be able to interpret it. And if they can't, they actually get a red light, yellow light, green light thing with
that's true. Right? Yeah.
And so it's a similar kind of process, but just with your genetics and genomics to just even as a person, you can get the red light, green light, yellow light. Everybody knows what that means. It's good or it's bad, or we got to keep an eye on it. So without having to understand what it really means.
Right. Well, and then there's the hopeful future you described, like 20 years from now, where I think we spoke about this in our last call, where I've had my whole genome sequence and, I've had it, interpreted with, different models of analysis. And one was, like the pharmacogenetics side of things. And, and then I have like an AI on top of it that I can communicate with. And so when I was getting a, surgery a couple of months ago, the anesthesiologist came out and said, okay, so, any drug allergies that you know about? And I said, well, honestly, like, no, because I haven't tried them all. Right. But your DNA kind of gives you a different filter. And so I can actually look at, and I mean, it's not perfect, but, it would say something like, oh, hey, you're a negative responder to Celebrex, for example. And actually, so there were no red flags on the anesthesia side of things, but there actually was on the outpatient protocol. So it's like, basically, if you want to have like a resting heart rate of 160, take Celebrex, right? And then I actually thought back to a previous surgery that I had, and I was like, oh wait, I actually remember that happening. I had an Aura ring, and I was like literally laying in bed with not 160 but 130, and I was like, I gotta get out of here. And I remember going for a walk in the winter, with my crutches, because I was just like having a panic attack in bed and not knowing the source of it. So I have the unfair advantage of having been blessed to think about DNA for 25 years. So, but even looking at your own DNA, it's hard to do it, objectively. And then even then looking at my daughters, it's it's hard to maintain that objectivity because, you're in a way you're emotionally compromised if there's an emergency on. So, you started to build a, community of experts around me, various practitioners, peer groups, because it really is, this whole notion of it takes a village. So like, how do you bring that kind of community notes, if you will, to how we, okay, we've talked about some, kind of maybe low, let's say minor, ethical issue, like, oh, well, should I know something that I can't necessarily do anything about? Is that going to cause undue stress? That's really on the educational side. Then at the end is like, okay, well, if we have a Canadian Genome Project, who the gatekeeper is to that data, right? And you get a lot of countries who don't want samples going outside of the country for, reasons of, well, let's just say, ancestry focus and biological warfare. I mean, I'm stepping into like conspiracy theory, et cetera, but it's not, it's not so far out, and it's not that people aren't asking these questions. And then there's the discrimination side of things. So for, Gattaca, I don't know if you remember that movie, that was like, well, you can't be an astronaut because, you don't have the genetics. So I don't expect us to kind of answer all of these questions, but I guess, a bigger question for me that I think maybe keeps me up is, how do you roll something like this out responsibly? And that is on the education, the communication, but it's also on the data warehousing and the data storage and the security, and like who owns this medical record.
Well, that's
for lack of a better word.
It becomes part of your medical records.
Right.
Right now, how secure are our medical records? Well, in Nova Scotia, they're still mostly hunks of paper or PDF files.
Yeah.
How are they stored and where are they stored? Australia actually did an audit of what's the best place to store healthcare records. Is it on a server at the hospital patient? Is it on, a hospital-based cloud that the hospital IT department is in charge of? Is it on a government cloud, or is it in one of the clouds where like the professional folks like Amazon or Microsoft have their clouds? And what is the safest one? Basically, the farther away you go from the hospital and the closer you got to the folks who do it for a living, Amazon and Microsoft, the safer your data was from being hacked. And so now Australia stores some medical data. So it's basically, you're moving to electronic health records. Well, it's going to be stored somewhere, somehow.
Yeah.
But, they're constantly they pay people too, and they're constantly trying to hack their own clouds all the time with the best in the world. And their core business is to make sure that it doesn't get hacked, or their business is hosted, right? And so in terms of storing data, it's a heck of a lot safer than somebody's laptop or server in the hospital or something like this, where you got like 3 IT guys trying to keep it from being accessed. So that's where Australia landed. And if your genome was just part of your medical record, it'll just be in there with everything else and protected as well as your current medical records are protected. So, Canada hasn't landed on anything yet. And we're a bit of an issue because we've got 10 provinces, 3 favorite locals. In some provinces, there's more than one health authority. Make their own decisions independent of each other when it comes to your own medical data, how it's stored, how it's protected. We don't have sort of a national data protection anything in Canada right now. What we do have is a law that is the Genetic Non-Discrimination Act that was passed a few years back. And basically that means nobody can access your genome. They can ask you if they can access your genome and give them consent. But if they try and access it without asking you, i.e. you had your genome sequenced as part of your medical care, and then an insurance company says, well, we want to access medical records to see what we need to, to insure this person or not, they cannot look at your genome. So they can't do sort of prognostic predictions. That said, they do it now anyways. I'll give you an example. I had colon cancer at 30. Now, I've sequenced my own genome as well. I don't have any of the genes that are known right now that predispose somebody to colon cancer. But where my son went to get life insurance, and they said, well, what's your family history, i.e. what are your genetics probably like? Not definitely like, but potentially like. And they said, we'll insure you for everything except colon cancer.
Wow.
Right?
Yeah.
So it still creeps in there. Now, the punishment actually in Canada for looking at somebody's genome without their consent is pretty big. It's $1 million and 5 years in jail. So we put a big punishment on it. So we do have the act in place. Nobody can look at your genome unless you let them. But you wouldn't let an insurance company take a look at your genome. It'd be silly. So that's here now. So there is that protection. The only way somebody could access your genome is if you give it to them, or if you're wherever it's stored. And it's wherever your medical record is where it's going to be stored. That said, I will tell you what, if I gave somebody my genome, my genome, or I gave them my wallet, which one would scare me more? Giving them my wallet. I'm way more than giving them my genome.
Yeah. Yeah. Right. Or any of your online passwords.
Yes.
Yeah. Yeah. It's kind of already all out there, isn't it?
It is. Yeah. So I think for some of this stuff, people are getting extra squirrely where they don't need to be. I don't know what's so special about a genome that's different from my blood work, that's different from my past doctor's appointment records, et cetera, et cetera. The only thing that your genome, and this is where the genetic counselors come in, is that, okay, you share genes with family members. And so there's a potential for other family members to have this same genetic issue that you've got, causing some sort of disease. And with genomics, we can actually then test. But what's one of the first questions any family doc or doc you see asks? Is there anything I should know that runs in your family? Is there a family history of blah? Which is basically asking, is there anything genetic, maybe environmental, but mostly genetic, that I should be concerned about? And your genome just gives you a far accurate answer to that question.
Yeah. Right. I guess the question is, well, I mean, it's a new tool, right? It's like,
it's a new tool. It can provide you yourself, if you choose to know, or at least your doctors, with something that is often prognostic. And again, we can say, for depending on the disease, sometimes it's absolutely, yes, you're going to get it. Sometimes it's you may or may not. And sometimes you're definitely not going to. So it's just a tool for you and yourself and doctors to actually have, so that if you do start to show weird symptoms, they don't have to start sending you all over the place to figure it out. They've probably got the answer in front of them already.
So we've kind of done this nice, weaving of your personal story as like the bright-eyed, bushy-tailed graduate student, to the let's say serial entrepreneur, and the North Star throughout is how do we reduce suffering, right? And you've probably dropped some. Well, you have dropped some nuggets along the way in terms of advice for aspiring scientists or entrepreneurs, or we just kind of went through the bigger picture.
Yeah,
I think what I'd like to do is maybe circle back to the questions that I mentioned on the outset, which I think it brings a nice summary to what we've discussed. And so those three questions were: what is it that we're saying really, and who has to hear it? As it relates to, let's not constrain it to genetics, but just maybe generally as we approach the underserved. And so that's number one. So what's the key message? Number one. And number two is who needs to hear it. And then number three is based on your experience, who would you nominate to be on the DNA of things?
So I think for me, in terms of big picture, medical research is in and of itself is meant to be collaborative. The end goal is to keep people as healthy as they can for as long as they possibly can. And if they end up either getting a disease or are born with a disease, what can you do to diagnose this as quickly as possible? And then what can you do to work towards a treatment that's as effective as possible? I think one of the issues with research, and this is partly on the way granting agencies work, federal granting agencies are the folks that give people money to do research, is they've turned it into a competition. And that's because they want to find the best research. They don't want to find a research that's foolish or makes no sense. Now, I'll tell you a story of my own experience when it comes to this. I don't think it should be competitive. I don't like giving people awards for medical research to say, oh, you're excellent and everybody else isn't. I think it sort of distracts from the big goal of everybody trying to work towards a common end. And so I got a large Genome Canada grant about 12 years ago. And somebody who I'd never met had gotten a large CHR grant, and it was to do the same thing. And we got them within a month of each other. And 2 months after that, we were both speaking back to back at the same session at a conference. And so it's like, okay, I outlined what our plan was, and she outlined an Mexican boycott. She's at the University of Ottawa. She outlined her plan. There was about 50% overlap, 50% different. And she said, my grant was for 4 years, her grant was for 2 years. She said, damn it, Chris, our plan was to use this as a jumping off point. And both grants were on rare diseases as a jumping off point to get, more money for rare diseases for our research. And I said, well, she says, now I don't think we'll be able to do it because we were going to go to Genome Canada or you just got your money. I said, well, there's 7,000 rare diseases. We're working on like 20. There's plenty of room here. He says, yeah, but if I propose it and it's the same thing on different diseases, I think they'll say, well, McMaster's already got some money. I said, I'll tell you what, I'll just give you my grant. You can read it. We just started the project. I'll give you my grant. You can read it. So that way you can propose something that's tangential or different enough that they'll say they're taking a new approach to study rare diseases. She said, what? I said, yeah, I'll just give it to you. And she says, okay, well, I'll give you my CHR grant and you can take a look at that. Maybe we can learn from each other about how we're trying to approach this. And we kept in contact. And then 2 years down the road, she wrote her Genome Canada grant, have it in my hand. And since we kept in contact, what's working on my end, what's not, what's working on her end, she wrote her really great grant, got a whap of money from Genome Canada as well. And I was actually on that grant. And it just, because they were taking a different approach to solving the same problems. And then it was turned into and that was about a $10 million grant. Mine was 5, the next one was 10. Then we worked together to get a innovation grant from CFI for infrastructure for our 2 institutions. It was a $20 million grant. Then we renewed the CHR grant. Then we did Genome Canada grant for another $12 million. And it was all because we decided we were going to work together rather than compete against each other. We could have decided at that very first meeting, you know what? We're both trying to work on rare diseases, and I want to actually ensure that I'm going to get all the money. But it's not about me. It's about the patients and the community, right? And so if you keep that as your North Star versus, okay, I just want to make sure that I'm successful, and so I want to get all the money for myself, well, then I never would have given her my grant 2 months into us having the grant, right? And so in terms of it working out for both of us from a career standpoint, it's been great. It's also way more fun to collaborate with somebody than compete against them. And I think we've got way more accomplished in terms of the science and money we brought in, to be perfectly honest, than if we would have competed against each other. And so I think there needs to be more of that type of attitude. I run a new PI meeting as part of my CHR job, and one guy stood up and said, well, my mentor at my university told me not to sign on to this large group grant that was being run out of another university because they're competitors of ours. I said, well, did you want to be on that other grant? Oh, yeah, it would have been great. I could have probably got so much done. I said, it's not about competition. It's about collaboration. They should be talking to each other and trying to find better ways to move the field forward rather than competing with it. And I think a lot of people, because you get a score, you want to get your next grant, you want to hide all your data from what you look at as competitors, but I look at as people should be collaborators. But that's why I also said no assholes allowed, because there's some people who they will just compete, and that's their mindset. And so I won't talk to or work with those folks. I think it's just a destructive way to go about the entire medical research energy.
Yeah. And it's
Question
Okay.
two.
Okay.
I've already forgotten because I bladdered too.
Well, who needs to hear it? So I think you kind of answered it. I mean, but if I was to paraphrase, sorry, go ahead.
Needs to hear it, but especially.
Yeah. Right. Well, I remember, like, as a grad student, it's like the worst thing that could happen to you while you're doing your PhD is you would get scooped on the paper that you were writing. It's like, no, actually, that might be a good thing. You've got a synergistic partner potentially. Yeah. Okay. And then the third question was, who would you nominate to be on the DNA of things?
If I was, it was the person I just brought up, because she's a medical doctor, MD, PhD, actually. It helps diagnose, and it's done I'd say she is Canada's expert when it comes to using genetics and genomics to diagnose rare disease kids. Probably one of the top 3 in the world. Kim Boycott of the University of Ottawa. Wonderful person, great collaborator. Kim is in KYM Boycott.
Sorry, her name is
Kim.
Dr. Kim Boycott?
Yeah, Kim is
KYM Boycott. Okay, gotcha. Perfect.
She's wonderful, gives a great interview, and she takes a different perspective because she actually sees the kids and treats the kids, diagnoses and treats the kids because she's MD, PhD, runs a medical genetics shop. She's the head of pediatrics at Children's Hospital Eastern Ontario, which is Ottawa's kids hospital. Top 3 in the world when it comes to this stuff. And as I mentioned my name, we still like each other. Actually, this is a side story, but we wrote this CFI grant for $21 million. It was her and I and one other guy. And their 2 schedules were so hairy that the only way we could meet long enough together to write this grant was to meet in London, England for 4 days.
Wow.
Well, I flew to London and they were flying from Europe because they had business in England up at actually the Wellcome Genomics Center, which is about an hour north of London. And so they said, well, we've got the 4 days between these. Can you come? And we sat in the lobby of the hotel 4 days running and pounded out a CFI grant, which got funded. $1 million. But it was, again, another story on the CFI grant was we all put in, the way CFI works is you put in your letters of intent, they publish the title, the person who's the PI of the grant. I put one in, Kim put one in, and another guy, Jacques, showed all put one in on infrastructure for rare diseases, essentially. So I called up Kim. I didn't know Jacques. Called up Kim and said, we both put a thing in. Do you want to work together on this? And she said, sure, makes lots of sense. It's worked out so far. And I said, you know the Jacques guy? She says, yeah, I know Jacques. Is he nice? He's very nice. Okay, well, let's call up Jacques and see if we want to put in a 3-way grant as collaborative instead of 3 people competing with each other. And we'll probably only get one funded on rare diseases for $7 million, $8 million. But if we put in all 3 of us, we might be able to combine them all and get her $21 million as this large national project and all get our money. And the whole field will then have $21 million for all of us instead of one person getting some. So again, it was an idea of let's not compete, let's collaborate, share each other's ideas, put the grant in. The fact that it was collaborative and it was national, we had a nice thing for diagnosis, biobanking, diagnosis through the screens, through the clinical trials. Because we were collaborating, we could put all that together. But any one of our sites could have done all those things together. So again, it was an idea of stop competing, just get along, play with nice people, and who have the same outlook that it's not about you. It's about trying to, diagnose these kids as quickly as possible, keep them healthy for as long as possible.
Amazing.
So for me, that's she's got a similar mindset. She's wonderful.
Amazing. Well, I mean, I'm recording this now, and I know that Rare Disease Day, Rare Disease Month is coming up in February. So, I mean, what a great time it would be to get your message out as, the world is trying to raise awareness on this topic. So, Chris, just before we go, what's the best way for people to find you? Because you really are somebody that, with gravitas. You are, attracting the underserved, and I'm sure there are people who are going to want to get in contact with you. So what's the best way to do that?
Jeremy, and whether I like it or not, if you Google Christopher McMaster, I'm like the first page of Google.
Okay.
And otherwise, it's email is the easiest. And again, it's Christopher.McMaster, and it's MC McMaster at DAL.ca. It's my work email.
Perfect. And I know you're on LinkedIn too, so I'll throw a link to your profile in the show notes. Dr. Chris McMaster, thank you so much with your time. You've been so generous. Thank you for being my guest on the DNA of Things. Well, thank you so much for joining us today on the DNA of Things, where we got this incredible insight, this notion that science is only as powerful as the system that governs it. And today, Dr. Chris McMaster showed us how genomics is moving from mystery to mastery, from research labs to real-world applications. And we explored how gene therapy can cure, not just treat disease, how scientific collaboration can accelerate breakthroughs, and why we must address genetic privacy and healthcare inequality now before it's too late. Now, if this episode expanded your perspective on genetics, medicine, and the future of health, please share it. Let's keep this awesome conversation going and expanding. Please tell a friend or family member, post your insights, and join us in shaping the future where science serves us all. Until next time, stay curious, stay informed, and remember, our genes don't define us, our choices do.